Okay, let's continue our discussion of Chapter 50 and look at the human eye. It's a fascinating structure with a covering called the sclera. That's the white part of your eye, although at the very edge here, where the cornea is, the sclera becomes clear and lets light through. You have the iris, the part of your eye that has different colors, green, blue, brown. Controls the size of the pupil, the opening into the eye that allows light through. Behind that, the lens, which is controlled by these suspensory ligaments, which are connected to muscles that allow the lens to change shape and for you to focus on objects at different distances. A liquid called the vitreous humor behind that, and on the inside of the eye, the part that's responsive to light is the retina containing the rods and cones and all the nerve cells connected to them that modify a light that we see into an image, into a nerve impulse, which is then translated into a image that we see, we perceive in our brain. Okay, so the optic nerve carries all these nerves connects all these nerves in the retina. The optic disc is where the optic nerve comes into the eye, and the orange part here, or yellowish orange, is essentially the retina. And you notice there's a gap here in the retina that at the optic disc, and that's known as your blind spot. That's the spot where the light that's coming through your eye that's hitting that spot, you will not perceive it. Okay, so the light is coming this way into the retina. The nerve cells are pretty much translucent, so the light gets through and it hits the rods and the cones, and they are the ones that have pigment that is receptive to light. In particular, they have this compound called retinol, which is embedded in a protein called opsin, and together that gives us rhodopsin. And this is what's changes in response to light. You can see it comes in two isomeric forms. In the light, it is one isomeric form, and in the dark, it's the other. And so these rods and cones, when light hits them, well, first of all, to say in the dark, they are polarized. They have a negative charge on the inside, but there's still some sodium ion that comes in. But when they are exposed to light, there is a process that essentially blocks these passageways, and such that the inside of the cell becomes even more polarized or hyperpolarized. And so that's what's happening to the rods and cones when they're in the light. So in the dark, when they are just plain old polarized, they release glutamate, and in the light, when they're hyperpolarized, they do not release glutamate anymore. You remember glutamate is one of these very common neurotransmitters. And so notice here in our retina, we've got the cones, and they're connected to these things called bipolar cells and also horizontal cells. The horizontal cells connect essentially different rods and cones and different bipolar cells. The bipolar cells are connected to the nerves in the optic nerve. They're also connected to these things called amacrine cells. The amacrine cells and the horizontal cells, they are said to essentially integrate information across the retina, such that particular rods and cones that are being stimulated by light, their information can be passed on to different bipolar cells and therefore different neurons. Alright, back down here. So in the dark, again, the rods and cones are releasing glutamate. In the light, they're not. And the bipolar cells that they are connected to, will receive that glutamate, and that bipolar cell itself can be depolarized or hyperpolarized depending on the kind of receptors it has for the glutamate. And the same in the light, when there's no glutamate being released, those cells can be either hyperpolarized or depolarized depending on the kind of receptors they have. So the bipolar cells essentially respond to different ways whether there's light or no light. And this is what allows us to see a complexity of information. Again, the lateral cells, the horizontal cells and the amacrine cells allow for information to be passed on to various bipolar cells and nerves in the optic nerve. And so this information is integrated. This is what's called lateral inhibition. And this allows us to see distinct edges on shapes and the contrast between images. And when fewer rods or cones are stimulated, we perceive this as a smaller field of vision, and again gives us a sharper image. And essentially it's because the light is hitting a particular part of the retina. If you look in the eye here, there's what's called the fovea. The fovea is the focal point of the eye. And at the fovea, the nerves there, the ganglion cells there, have a very, very fine and small receptive field. So that's where essentially you see the best, and have a very sharp image at the fovea, or the focal point of the eye. You can perceive images on the rest of the retina, but they're not quite as sharp as that area. And so that's when you're looking forward, or whatever it is you're focusing on, that is the image. The light from that image is going to be hitting on the fovea, and the light coming from other images off to the side aren't hitting the fovea, so that's why they're not quite as in focus or sharp an image. And of course, if you want to make those things have a sharper image, you have to look at them directly to get them, that image hitting that focal point, that fovea. Visual information. So of course we have two eyes, and there's light from objects coming into both the eyes, but they're hitting different parts of the retina and stimulating different nerves. And so the optic nerve from each eye connected what's called the optic chiasma, and some of those nerves essentially cross from left eye to the right side of the brain and the right eye to the left side of the brain. The information is passed into the primary visual cortex of the occipital lobe at the back of the brain, and that's where we are processing a lot of this information, making associations between what we see and what we know about what we're seeing, where our memories are of these images that we're seeing, and we're able to make sense of them. Okay, vision, very complicated. Let's go to taste and smell. Gustation is taste, olfaction is smell. We use our tongue to taste things. We have these taste buds inside our tongue, these little pores with all these cells that are connected to neurons, and they are receptive. Within the taste bud, there's different of these receptor cells that are sensitive to particular kinds of tastes. So depending on what chemical is in your mouth, you will perceive it as a different kind of taste. And with smell, there'll be compounds in the air that get into your nasal passages, and your nasal passage is lined with mucus that have these nerves embedded in them, and so as those compounds get stuck in the mucus, they will bind to those cells, and they will stimulate nerves in your olfactory bulb that are sent to the central nervous system, and you can process that particular information, those particular smells. Okay, now this section, muscles. So of course we've talked about muscle tissue a little bit, but let's talk about it in a little more detail. So of course we know muscle is made largely of protein, and there's two main types, what's called actin and myosin, and they come in these overlapping filaments, and it's the sliding apart and together of those filaments that allow muscles to relax and contract. The basic unit of a muscle is called a sarcomere, again with these overlapping actin and myosin filaments. A myofibril is a bunch of these sarcomeres, one after the other. A bunch of these myofibrils make up a muscle fiber. A bunch of muscle fibers make up a bundle of muscle fibers, and when you put a bunch of the bundles of muscle fibers together, you get a muscle. So we've got this nice hierarchy here in the structure of muscles. The contraction happens when essentially the sarcomere gets shorter, and those actin and myosin filaments are sliding past each other. This is a process that takes energy, of course, and at the cellular level that source of energy is the ATP. And essentially the myosin has these little extensions that attach to the actin filaments and allow the myosin to sort of move along the actin, causing the sarcomere to get shorter and causing a muscle contraction. We control these processes, the contraction of muscles, with our nervous system, and so you have nerves that are connected to your muscle, and they release neurotransmitters that then cause a change in this portion of your muscle that sort of surrounds those sarcomeres, called the sarcoplasmic reticulum. The impulse from the nerve causes the sarcoplasmic reticulum to release a bunch of calcium into the area around the sarcomeres, around those actin myosin filaments, and that essentially stimulates the nerve impulse. And you can see it's the ATP that allows for the movement of these calcium ions. You have, so in a muscle, you have a bunch of nerves, a bunch of motor neurons that feed into that muscle. And so they're in different units. So one neuron will feed into some of these muscle fibers, and another neuron will feed into another, etc., etc. And this allows you to have various levels of muscle contraction depending on how many neurons, motor neurons, are being stimulated. So as you've experienced, when you pick up something that's very light, it doesn't take a whole lot of muscle power to move that thing, as opposed to if you were to pick up a bowling ball, and it's going to take a lot more muscle. So your body is able to quickly perceive what it is, how heavy it is, the object that you're picking up, and with very light objects, just some of your muscle fibers will be stimulated, because you don't need to use all of them, because that would be wasting energy. But if you're picking up a very heavy object, more of these muscle fibers will be stimulated to contract, allowing you to pick that heavier object up. Okay, this is a long chapter. We're going to get to section 6 in another video.